Hi dear students, today we're going to talk about RNA transcription in the case of prokaryotes. And you know what is RNA transcription? RNA transcription is a process by which mRNA is being synthesized from DNA. And it is later on that we can see that this mRNA is being further utilized in the protein synthesis. So the process of synthesis of mRNA from DNA is referred to as a transcriptional process. Now this is a seminar which is being conducted by Alina, and after her seminar I will be giving a feedback on that.
Good afternoon, friends. Today we are discussing transcription in prokaryotes. Before that, we have to know what is transcription and where does it occur. What is transcription? The process of synthesis of RNA by copying the template strand of DNA is referred to as transcription. It can also be defined as the synthesis of RNA from DNA. In prokaryote, transcription occurs in the cytoplasm. We have studied transcription and translation simultaneously occur in prokaryotes because they don't have a true membrane-bound nucleus. RNA polymerase is an enzyme which is involved in transcription. And this RNA polymerase can initiate transcription by itself and it doesn't require primase for initiation. ATP, GTP, CTP and UTP are used to produce an RNA complementary to the DNA template. RNA synthesis proceeds in a 5' to 3' direction with new nucleotide being added to the 3' end of the growing chain. RNA polymerase is an enzyme involved in transcription. I have already said. It doesn't require primase action in the 5' to 3' direction. Most bacterial polymerases contain 5 types of polypeptide chains. They are alpha, beta, beta-H, sigma and omega. The core enzyme which catalyzes RNA synthesis is composed of 5 chains, 2 alpha chains, beta, beta-H and omega. The sigma factor has no catalytic activity but helps the core enzyme to recognize the start of genes. When sigma is bound to core enzyme, the 6 subunit complex is referred to as RNA holoenzyme. This RNA holoenzyme only can begin transcription, but the core enzyme complete RNA synthesis once it has been initiated. The precise functions of the alpha, beta, beta-H and gamma are not yet clear. A prokaryotic gene structure is clearly depicted in this picture. In this figure, you can see a promoter region, RNA coding sequence and a terminator region. The region 5' of the promoter sequence is called upstream sequence. The region 3' of the terminator sequence is called downstream sequence. What is promoter and what is its significance? A promoter is a sequence of DNA to which proteins bind that initiate transcription of a single RNA from the DNA downstream of it. Promoter is nothing, the DNA sequence onto which the proteins and enzyme involved in transcription binds and that initiate the process is called promoter. Bacterial promoters have 2 characteristic features. A sequence of 6 bases, often TTG-ACA, about 35 base pairs before the transcription starting point, and a TA-TA-AT sequence or Pripna box, usually about 10 base pairs upstream of the transcriptional start site. These regions are called -35 and -10 site respectively. You don't have to confuse what is -35 site and what is -10 site. The -35 site, which consists of 35 base pairs, which is present before the transcription starting point, and the -10 site, which consists of 10 base pairs present in the upstream of the transcriptional start site, comprise the core prokaryotic promoter. RNA polymerase holoenzyme recognizes specific sequences at the -10 and -35 site of promoters. These sites must be similar in all promoters and they are referred to as consensus sequences. Once the RNA polymerase bound to the promoter site, it is able to unwind the DNA without the help of helicases. The -10 site is rich in adenine and thymine. In DNA, adenine always pair with thymine by 2 hydrogen bond. This hydrogen bond that keep the DNA double-stranded. When the DNA unwound at this region, it is called open complex. The region of unwinding is referred to as transcription bubble. I'm going to explain steps of transcription. Transcription consists of three major steps: initiation, elongation, and termination. In this figure, you can see minus 35 sequence and minus 10 sequence, what I had said in the previous slide. The first step of transcription is initiation. We have already know, during replication, the entire genome is copied. But in the process of transcription, only a short segment is transcribed. When the RNA polymerase bound to the promoter region, the initiation begins. But this RNA polymerase is not able to bind the DNA tightly. The situation is drastically changed when the sigma bound to core to make the hollow enzyme which bind the promoter tightly. The promoters serve only as a target for the binding site of the RNA. This promoter is not transcribed during the process of initiation. Then the sigma factor of the RNA polymerase hollow enzyme is responsible for positioning the core enzyme properly at the promoter. The sigma factor recognizes two regions in the promoter, one centered at minus 35 and the other centered at minus 10, what I have said in the previous slide. Once positioned properly, the DNA at the minus 10 region unwinds to form an open complex. This open complex is clearly depicted in the picture. You can see that. The sigma factor, after unwinding, then the sigma factor dissociates from the core enzyme after transcription is initiated. The second step of transcription is elongation. The region of unwinding is referred to as transcription bubble. The transcription bubble moves with the RNA polymerase as it proceeds to transcribe mRNA from the template DNA during elongation. Within this transcription bubble, a temporary RNA DNA hybrid is formed. As the RNA polymerase progresses in the 3' to 5' direction along the DNA template, then the sigma factor soon dissociates from the core RNA polymerase and is available to aid another unit of core enzyme to initiate transcription. And mRNA synthesis happens in the 5' to 3' direction, and newly synthesized mRNA is complementary and also antiparallel to the DNA template. As elongation of the mRNA continues, the newly synthesized mRNA is released and the two strands of DNA behind the transcription bubble resume their normal helical structure. In this figure, you can see the process of elongation. The region of unwinding and RNA DNA hybrid is clearly depicted in the figure. You can see that. The last step of transcription is termination. RNA synthesis will continue along the DNA template strand until the polymerase gets a signal that tells it to stop or terminate transcription. Termination occurs when the core RNA polymerase dissociates from the template DNA. The end of a gene or group of genes is marked by DNA sequences in the trailer and the terminator. Sequences within the prokaryotic terminator often contain nucleotides. When these nucleotides transcribed into RNA, these nucleotides form hydrogen bond within the single-stranded RNA. This base intrastrand base pairing creates a hairpin-shaped loop and stem structure. Once the hairpin loop and stem structure appears, that indicate or tell the RNA polymerase to stop or post-transcribing DNA. There are two kinds of terminator present in prokaryote: intrinsic or RHO independent termination and RHO dependent termination. Coming to RHO independent termination or intrinsic termination. In intrinsic termination, RNA first form a hairpin structure. Once the RNA polymerase has posed at the hairpin loop, the AU base pair, that is adenine and uracil base pairs in the uracil region, are too weak to hold the DNA RNA duplex together and the RNA polymerase falls off. In intrinsic termination, RNA forms a hairpin structure which displaces RNA polymerase and stops transcription. Coming to RHO dependent termination. This kind of terminator lack a polyuregion and sometimes it lack a hairpin structure. It requires the aid of a special protein referred to as RHO factor. This terminator causes RHO dependent termination, and this RHO bind to mRNA and move along the molecule until it reaches the RNA polymerase. This RHO factor also has a RNA DNA helicase activity, then it causes the polymerase to dissociate from the mRNA, probably by unwinding mRNA DNA complex. In RHO dependent termination, it need a special protein called RHO factor. Then RHO bind to the mRNA and move along the molecule, and this also has a DNA RNA helicase activity. Then it causes the polymerase to dissociate from the mRNA and probably by unwinding DNA complex. This is a pictorial representation of RHO independent termination. In this figure, you can see newly synthesized mRNA. The red color shows newly synthesized mRNA. In that, you can see a hairpin loop structure. While the RNA polymerase poses at the hairpin loop, it is not able to hold the RNA DNA hybrid together, then termination occurs. This is a pictorial representation of RHO dependent termination in prokaryote. In RHO dependent termination, RHO protein comes and then bind to the mRNA and move along the molecule until it reaches the polymerases, and it has a RNA DNA helicase activity. Then it will cause the polymerase to dissociate from the RNA, probably by unbinding the mRNA DNA complex. Again, I repeat the process of transcription for your clarification. Transcription consists of 3 steps: initiation, elongation, and termination. At initiation, when the RNA polymerase bound to the promoter region, initiation process begins. Then, the sigma factor of the RNA polymerase holoenzyme is responsible for positioning the core enzyme properly at the promoter. Sigma factor recognizes 2 regions in the promoter, one centered at -35 and other centered at -10. Once positioned properly, the DNA at the -10 region unwinds to form an open complex. Then the sigma factor dissociates from the core enzyme after transcription is initiated. Coming to elongation, the region of unwinding is referred as transcription bubble. The transcription bubble moves with RNA polymerase as it proceeds to transcribe mRNA from the DNA during elongation. Within the transcription bubble, temporary RNA DNA hybrid is formed. RNA polymerase progresses in the 3' to 5' direction along the DNA template. The sigma factor then dissociates from the core RNA polymerase and is available to aid another unit of core enzyme to initiate transcription. The newly synthesized mRNA is complementary and anti-parallel to the DNA template, and newly synthesized RNA is released, and the 2 strands of DNA behind the transcription bubble resume their double helical structure. Coming to termination, RNA synthesis will continue along the DNA template strand until the polymerase gets a signal that tell it to stop or terminate transcription. There are 2 kinds of terminators: intrinsic or RHO independent termination and RHO dependent termination. In RHO independent termination happens when the RNA form a hairpin structure which displaces RNA polymerase and stop transcription. In RHO dependent termination, it needs a special protein called RHO factor. This RHO factor binds to mRNA and move along the molecule until it reaches the RNA polymerase. This RHO factor also has a RNA DNA helicase activity. Then it causes the polymerase to dissociate from the mRNA, probably by unwinding mRNA DNA complex. I hope this is clear. Thank you.
Hi dear students, I hope you got a good understanding of what is transcription, but I need to make a correction which was told by Elena. We had told that from the transcription start, -10 base pairs before that, there is a site called the Tata site, and -35 base pairs before the transcription start point, or upstream the transcription start point, we have the TT Gaza site. So it was, I think, a correction, like she had made a mistake that she told that it is minus 10, it is consisting of minus 10 base pairs and it is consisting of minus 35 base pairs, but that is not the thing here. You should understand, if you have a particular line, now if consider this is the transcription start point, okay? If I go forward towards the transcription site, that is ahead, okay? And if I go behind this, that is behind or before the transcription start point, before the transcription start point, minus 10 base pairs before that, there is a site called the Tata box. And this is the transcription site, and you go minus 35 base pairs behind. Okay, that is before the transcription start point. That is referred to as a TT CASA site. So I guess this is clear to you. I just wanted to make a small correction. The remaining things are fine. Thank you then.